So with that, let me introduce the next speakers. So the next talk focuses on AI for synthesis planning, all this predictive modeling that we have heard for off-target prediction or target prediction. Now we talk about synthesis planning. And the first, we have two wonderful speakers. Bart, good to see you again. So Bart is a senior scientist at Boehringer Ingelheim. He works as a medicinal chemist on various drug discovery programs for both human pharma and animal health. Bart holds a PhD, and he has done previously. He has done research at Bayer Crop Sciences and a postdoctoral stay at MPI KOFO. Quite a lot of acronyms there. I'll have to look it up. Thank you for joining the conversation, or thank you for being here to give the talk. The next speaker alongside Bart is Max Boehm. He's a chemistry solutions specialist at Reaxys at Elsevier. He works with customers in the Western US and Canada across various industries on how best to use Reaxys in their research. Before joining the Reaxys team, he worked at TCI Chemicals in product management and sales. He holds a PhD in chemistry from Technical University of Denmark. Thank you both for being here. And with this, I hand over to you, Bart.
Thank you very much, Abhinav, and thank you everybody. Sorry, thank you everybody online for joining in. First, I want to start by thanking the Elsevier team for the organization and inviting me to speak here. Thank you very much. It's much appreciated. The slides that I'm presenting are prepared by me and a colleague, but unfortunately, I have to present them today by myself. Before I get started on sort of the real content, maybe for the people who don't know, Boehringer Ingelheim is a relatively big company, one of the top 20 leading pharmaceutical companies in the world with about 52,000 employees. I won't go into further details on these slides. I just wanted to highlight that we have several research sites across the globe, and the project that we are doing and which I will be presenting here, so the predictive retrosynthesis tool, of course, mostly applies to chemists, which we have then in Ridgefield, Biberach, and Vienna mostly, but also the Ingelheimers can, of course, use it. Sort of an introduction to what we want to do and why we think it's important and how we position this tool at BI is what I want to walk through in the next few slides. So what you see here is kind of the classical way I think most people remember from the bachelor, master, PhD, or maybe even their job experience, is that chemist has a nice idea, wants to synthesize a molecule, and then you go to your favorite user interface, and as this is the Reaxys Global User Day, I would say let's go to Reaxys. Then you use that user interface to search the database for reactions that are maybe roughly similar in structures, for example, and from that you get some literature. Then the chemist needs to read and evaluate the literature. Maybe it's not good enough, you go back in the process and you go through this cycle several times. This is a little bit time intensive. It can be very enjoyable, but can also be very tedious, but at least it's better than going to the library. And now we're going sort of in the next wave or the next type of literature search. And that's the computer-assisted synthesis planning as we see it, at least at Boehringer Ingelheim. So what we want to do is outsource these iterative searches and comparison to a machine. Then this tool, the CASP tool, will just browse the database and can extract all the related data and structures for you, and then index this, for example, by structural similarity or reaction similarity. And this could save a lot of time. Then it, of course, can give this back to the user interface, and you can read it and through there download the literature examples. And this is super nice, but this is then limited to reactions that, for example, are already known or substrates that are already known. Taking this then one step further, you could also have then an AI-powered retrosynthesis tool to generate a synthesis proposal on molecules that haven't actually been reported, and then also provide you ideally with literature to those, or at least literature very close to what you're seeing on the screen. And then finally, taking this one step further, as a business case for us, is that it should also connect, for example, to the stockroom. So your building blocks, you can see that they are available at your site. You can just order them to the lab and start your chemistry essentially right away. And also ultimately tie this into your electronic notebook. So what this is, or how we see it at BI in summary, is CASP as a tool or a smart assistant, if you will. So not the ultimate problem solver that can crack the really hard molecules. It's more maybe like somebody you could compare it to, maybe a bachelor student that reads the literature and compiles a report for you. This is, in our opinion, sort of the next phase in literature searching. So we went from the library to databases that are online, which is already saving us loads of time. And now we're sort of ready for the next phase, which is then CASP. And what is especially nice in doing this is that you can also already check, for example, for building block availability, or as I'll show later, you can integrate this even further into your IT landscape. And what we want to achieve with this is essentially liberating time by letting the machine do the sort of like easy steps, and letting us, for example, me as a head of a laboratory, have more time to actually focus on the more difficult problems or the difficult syntheses. And all in all, we focus this as a medchem tool, and we want to increase the speed in medchem in the discovery process. So a little bit of history on how we got to Reaxys RT at Boehringer Ingelheim. We founded the CASP team back in 2018. That was shortly before I joined the company, actually. But I joined the team essentially as a tester, and quickly took over the lead of the team. What we did straight from the beginning, obviously, is monitoring what is out there. We set ourselves the goal to not develop it, but to license something and bring something in-house, gain experience with this. And that's how we started. We found then in 2019 a first sort of commercially available tool that we could use. So with first a few people, up to 10, sort of the core team, we evaluated this and then decided to run a pilot with some what we call the early adopters. So people that are open to this technology, more or less 50 people globally. And use this tool for a year, get experience, get feedback, give feedback to the company that rolled it out. And we did that from the end of 2019, basically to the end of 2020. But then also in 2019, Reaxys came out with a beta version of Reaxys RT. And we tested that simultaneously in the small team, and we actually really liked it. And it also immediately revealed maybe some of the things for our business case that fit better than the tool we were already testing. Nevertheless, we conducted the tool. Reaxys at that time was a beta version, so we couldn't really do anything with that. So there's a little gap between 2020 and 2021 when Reaxys came up again to us and said, well, it's ready for you to test it as a real product. So we did that. We started in 2021 with the pilot, which ran until the end of 2022, essentially. And in the meantime, what we also did then in the middle is a head-to-head comparison on all the tools that were then available at that time. And I'll show a little bit more details later. But out of this pilot, we basically concluded that Reaxys was at that time, and still is, the best fit for our business case. So really speeding up the discovery process, the early research. So 2023, we licensed Reaxys RT, and in February, we rolled it out globally. And for those who get the joke, we started in Biberach, therefore there's a beaver at that time point. So to go a little bit more in detail, like on sort of for the scientists, like comparative details and showing some graphs, we have here, for example, we looked at the quality, and what we did a sort of an Amazon style rating. So 1 star being not so good, 5 stars being essentially something you can give to the lab and perform right away without further modification. 4 being good enough, maybe for a skilled person to make minor modifications, but the general idea is there. Then 3 is something that, yeah, it could probably work. You wouldn't do it, but like it would maybe not fail. And 2 would then be already not a good proposal, and 1 would be really something that's really off and doesn't really make sense. And we saw in quality that Reaxys slightly outperformed the other tools, although also the other tool, which let's say CASP2, performed over 50% basically with satisfactory results. When we then looked at diversity, we see that it's roughly similar for most of the tools, so more or less 50%, if you will, is satisfactory, and the other 50% was really not so good. And with diversity, I mean, let's say you're looking at a cross-coupling, you can do it with an aryl bromide, and then the next proposal is an aryl iodide, and an aryl triflate, and an aryl iodide. It goes on. This, of course, is not very helpful for the synthetic chemist to see all these options. And maybe one note here is we looked at diversity without any post filters. So this is really as it came out of the tool. We also wondered how important this is in the end for the end user, but at least for us, is one of the parameters we took into account, where most tools roughly performed similar. And then the next one, which is quite interesting, is the reference quality. And you can see here in the graph that there's basically two tools that performed really well in that aspect, and really over 80% high ratings for reference quality. And if you then look at the next graph, we found out that for the user satisfaction, this might be the driving factor, because I think we all know, we're all chemists, and we all see that we're used to this process of going into the literature and then quickly deciding if this is maybe feasible or not. We're quite good at that process. So if you provide a spot on reference, we can very quickly see if it makes sense for what we want to use it or not. Even if the quality is not so high, let's say many of the results you get are maybe not good, but one or two are, you can quickly see this as long as the references are really good. And this is exactly what we saw here. So Reaxys outperforms basically any other tool because of the combination of quality and reference, the reference quality. So that was one, the main driving criteria in quality wise and as like user satisfaction. But there were also some additional advantages to Reaxys, but we found this the best fit. And sort of the additional advantages to Reaxys, the things that we liked, at least in the team, were that these new features were added to the existing Reaxys homepage. Most of us were based in Europe, we're quite familiar with Reaxys. It's very intuitive, and also the Reaxys RT tool was very intuitive to use. So we found that we didn't have to train people on how to use it. Additionally, we were able to at least in a simple version and quickly integrate this into our existing IT landscape, and I'll show some examples of that a little bit later. And the other thing that I think is really important, considering all these tools that are currently out there, is that this tool is based on the underlying Reaxys data. So you have really high quality data and lots of it, which I think, in my opinion, makes this tool much better than many of the other tools that are maybe trained on only publicly available patent data, for example. And hopefully it's also self-learning on new data. As new papers come out and they get indexed by Reaxys, they will be added to the database and we'll be able to find them. And finally, we have as a company been involved in these discussions very early on, and we've also consulted on the user interface. So I think that also makes this product now a very good fit for us, as we were able to shape it a little bit together with Reaxys. So here's now sort of the overall picture where I've sort of highlighted in a lighter color what we have achieved, and I think that that's all well on its way. So we have integrated our building blocks, we have integrated, or our building blocks at least connected to our ELN, and also the design studio, which would also connect then to the ELN, is connected with CASP somehow, and I want to show that in the next few slides in a bit more detail. So us designers, chemistry leads, we use this design tool where we can draw certain structures and then check for calculated properties. For example, we can check how structures relate to in their SAR before actually synthesizing them. One of the things where we also assess there, of course, is the synthesizability of these compounds. And now we can basically with one click send this structure to Reaxys. The search automatically starts running, and we will see on one hand if it has published routes immediately, which in our case for drug discovery is maybe not the best thing to see. But then other side, you can see also the predicted routes, which then gives us quickly a feeling on how easy or difficult this molecule might be to make. And since we have usually lots of designs to go through, this is very valuable for us to maybe, if you, let's say, have 10 designs, but you only have time to make 1 or 2, here we can assess which one would be then the first ones to make. Well, synthesizability is one of the parameters, but it's definitely helpful if you can get a compound very quick to test a certain hypothesis. And then the last feature that we really worked on before rolling it out at BI is the direct ordering of the building blocks and also seeing the compound availability. So it's highlighted here. Apologies that it's a little bit small, but there is always this little shopping cart under the structure in Reaxys. And at least if you work at Boehringer Ingelheim, you can click on this. You see the commercial suppliers, obviously. Then at the bottom, there's also this custom availability, it's called here, because it was still during the pilot phase. And you have a Boehringer Ingelheim logo. And if the compound is available, this would appear. If the compound is not available at Boehringer Ingelheim, this would not appear. So you could immediately know whether we have it or not. And if you click on the logo, then you would take you to our proprietary inventory system, and you could order it to your lab basically the same day, which would also really help speeding up the chemistry. So when we rolled out, the Reaxys tool, we said there are certain must-haves, so those have to be completed. And there are certain things that we would like to have. And as you can see, the must-haves, we wanted to integrate this in the design tool. This is to get really the design teams on board and start using it. And we wanted to have the compound management tool integrated so that also the people that work in the lab do the synthesis, they can actually save time just by simply using it. Another thing that we're still working on, and together with Elsevier, is to have this unique link that you get for every Reaxys RT search, to get that pushed back into our design tool, so other users also can open it from there and don't have to then restart the search for each individual user. And then there are some open user stories. So computational chemists, they design even more molecules with a computer than we do. So we're not talking maybe 10 or 100 molecules per design team meeting. We're talking 10,000 to 100,000 molecules. And they have to be ranked, of course, in many ways to trim that number down to make it actually possible for the synthesis labs to get through those. And one of the things that we want to do is maybe synthesizability again. Currently, the calculations take 2 to 20 minutes. This is fine for tens, maybe to 100 molecules. But this takes very long if you start doing 100,000 molecules. So that is something we're working on with Elsevier, trying to improve that and fit that story into a product. And of course, full and seamless integration with the ELN and our IT landscape. This is completely on our side, I would say. So the new APIs look very promising, but development on our IT side is not as simple. And I'm just a medicinal chemist, so I don't really influence that. The switching gears a little bit. So we have the tool. This was all very technical. This is how it performance. But I want to talk a little bit about how we rolled it out and how we integrate people basically with the tool. And one of the things we do is we also use it as an education tool to get people familiar with it, but also to work on their education. And for those who are not familiar with the German setup, many bench chemists are only have a vocational training, so they are not necessarily bachelor, master, or PhD chemists. They're very good in the lab, and we are trying to train them on maybe more synthesis knowledge or like theoretical knowledge, because they're mostly working based on lab experience. So we do this, for example, with training by the supervisor. They have worksheets where they work on retrosynthesis. There are, of course, seminars. And also they could get a bachelor, for example, on the job. What we think is that CASP, and so the retrosynthesis tools, they are very convenient in accelerating their development, because it's generally easier to recognize a good idea than maybe come up with your own all the time. And so what we do is we have then in these, for example, the worksheets and the retrosynthesis exercises that we do in the company, we will also include these retrosynthesis tools where people can just look up the molecule, criticize or scrutinize maybe the proposed routes, and have maybe the discussion also with the supervisor saying, hey, I like this route, what do you think? And maybe the supervisor can ask back, okay, why do you think it will work or will not work? That's one of the things we want to do. And at the same time, we familiarize people with the tool, which we hope then makes it also easier to do their job. And to get sort of the adoption strategy we looked at, and we thought about this hard because we've always faced sort of this catch-22 with this tool when we were doing it, when we were going basically from the innovators. So that's basically in this case, the core team that is sort of has been interested from the beginning, wanted to get some early adopters on board. Generally, these people are quite interested already. Then you get this catch-22 when if the tool predicts something they already know, say, well, I don't need a tool because I already know it. But then if it predicts something they don't already know, they say, well, the tool must be wrong because I don't know. And this is something we say, okay, how do we best deal with this problem? Again, this is mostly talking with my other lab heads. And we went for a grassroots strategy. So basically we started at the bottom and work our way up the hierarchy tree and not going top down. So we focused initially on the innovators. That's kind of easy to find. That's us. Who are driving these initiatives. Then we focused thereafter on getting the early adopters on board. These are usually people that already have maybe a knack for IT or like to play with new tools or are very good at synthesis and just want to expand the horizon. And then from that push into the early majority and have that diffuse. And our goal is to stop somewhere at the end of the early majority, late majority. We do not want to focus on getting everybody on board by force, essentially. So that's also summarized here a little bit. So we want to get initially the motivated and the excited people. And where do we find them? Well, they are, for example, taking already the internal classes. They're already participating in courses. They are playing around with other tools. So they're quite easy to identify. Then we start with them. Then, as I said, we go into the worksheets. And maybe we have to rethink a little bit how we do that, and we include Reaxys RT in that. And we have also integrated there in our retrosynthesis exercise, seminars that we do. And then once we have that established, and we have this group of early adopters, which we also use for the pilots, essentially. We start using them as influencers because they are usually people that are very good at chemistry. They're very excited about it. So they have actually quite a large influence at the teams. And we really strongly focus on the people in the bench because we feel that those people have the most benefit. Not only helps their chemistry, but also the ordering of their compounds. And then what we've seen now is that then the proposals they go to the supervisor with, they come maybe from this tool, and then the supervisor starts using it. So I think this is overall working quite well. I think we have more or less some regular users from about half of the people in the department, which isn't the perfect number, but it's pretty good. And then finally, I want to conclude and also look a little bit at what made this a success and what not, and what is sort of the outlook. So we successfully compared, I would say, all the existing tools that are available commercially and have picked Reaxys out of that as the best fit, at least for our use case, which is really accelerating medchem and discovery chemistry. And what we need for that is a very strong IT support, we found, because this, at least at Boehringer Ingelheim, we've driven this really from medchem. So this is really mostly lab heads and other bench chemists that are very excited about chemistry, but we're not necessarily IT experts. So we have the dedicated team there and IT support. We had very good communication with Elsevier, very pleasant to exchange, and they've really listened to our feedback very openly and also adapted maybe the tools in ways that we said that could be beneficial for us, and also close exchange with other initiatives on IT tools in our company to make sure, on one hand, we don't all do the same, but also maybe finding synergies and maybe also feeding that back to Elsevier and seeing what we can do in the future. Then a very important thing was to have certain systems already in place when we roll it out. We felt we can only make one wants a first impression, and it has to be a good one. So we said we do want to make sure that we have, for example, the compound management tool in place, that people can immediately use it and start ordering reagents. This was quite a lot of work, and we were constantly also in a dialogue with Elsevier to make this happen. Yeah, and finally, I think it was very important to start with the grassroots strategies, to really start from the people that maybe most benefit from the tool and work our way up through the design teams. And I think with that, I want to close, but not before I show you this slide, which is also for download. So if you like what we do at Boehringer Ingelheim, you can take a look at these career offers or postdoc offers, or in case you're not familiar yet with our OpenMe platform, please check that out. And thank you very much. And I'll take questions after Max is done with his part of the presentation. Thank you.
All right. Thank you so much for this great overview. So we've seen now how Reaxys predictive retrosynthesis can be used as a real time saver in the lab to compile literature information instead of having to manually look through different reactions. You can get it compiled by the machine. You'll be able to focus on the more challenging aspects of your job, and the educational aspect was also very valuable. What my goal is for this presentation is to just illustrate what that workflow actually looks like. How would a chemist go through this, make use of it, find the information, and how can that impact their workflow? So what you see right now is the front page of Reaxys. This is the place, if you're not familiar, where you would submit your usual searches for structures and reactions. We've seen earlier the query builder, where you can perform more advanced searches. And then we have the retrosynthesis section. So I'll go straight into this. And what you'll see is a table that lists all the retrosynthesis projects that you've submitted. You can name them. You can sort them by either the name or the date of last modification. You'll see the structure, and you'll get the overview of the different predictions that have already been generated. Now, to create a new query, you would just click here to draw a new structure, which opens up our structure editor with all the usual tools. I'm going to just insert a smile string to make it quick. And what you see now on the right-hand side is a few sets of parameters that you can use to direct that prediction a little bit. We can see there's one section for prediction, there's one section for published. So for those who are not super familiar with Reaxys and published, this will generate the similar style route as the prediction in terms of the user interface, but it's only going to be based on exact literature precedent, which means if this structure is not documented in the literature, you will not find any hits from your published search. So this is where the prediction comes in really handy. And we can take a quick look at the parameters here. We can choose between a full route prediction or a last step only prediction. Sometimes this can be really interesting for more challenging tasks where you would want to find a potential entryway out of multiple ones to start your retrosynthesis. You can modify the diversity of routes by configuring the reusing of reaction steps or building blocks. So if you imagine you plan your synthesis, you come up with different disconnections. Depending on your situation, you may be able to reuse the steps or building blocks you identified in a different order. And so this will allow you to do that in the prediction. We can choose between two processing time settings. There's a standard time, which will be up to 10 minutes, and the extended time that gives you up to 30 minutes of calculations. So again, when you have a more challenging molecule, this is one of the parameters you want to play with. Now, really important, we've already heard from Bart about the building block library integration. We do have our own building block library, which is quite extensive, and you can see different categories of those building blocks. If you were interested in using your own library, we would be able to add another setting there to make sure that that's included as well. And so this is important because all building blocks that are starting materials in the predicted routes will have to be part of this set of the libraries. We try to make sure that the materials that you need to start with are actually somewhere available. Now, once you've set your parameters, and usually you would do this once as a default and then just submit every new molecule there, and only if you run into some more difficult projects would you come back and modify those settings. So this is running right now. We can see there's nothing in the published literature for this. So this will take up to 10 minutes to calculate. And meanwhile, we can take a quick look at a different example that I've prepared. Let me quickly pull that up. So we can go into this one. What we see here now is the big picture overview. We saw that there are 5 predicted routes, and those are listed here row by row. And then for each route, you will get the list of starting materials, the number of steps, an overview of what that route looks like. Is it more linear, more convergent? And in the case of a prediction, you will also get this confidence score, which expresses how good of a route this would be. And usually you can see it starts here at 0.95, and that score can drop, in this case, up down to 0.6. Now, this is really useful view because you can immediately see something about the commercial availability for each building block. So we have the shopping cart icon here. If you just hover over it, you'll get a brief summary of the available data on that. If you were to click on this, you could go into our commercial supplier database for this particular compound, which will give you a table of all the suppliers we have, which again, you can filter by all the different categories like price and size and all these things. And you can obviously also go into the supplier's website to order it or check on the most current status. Now that we're here, we probably want to take a look at this route. So we would click on the tree view, and the tree view will consist of 2 things. We have the main window here, which is the full route starting on the left-hand side with the first steps all the way to the final step to your target molecule. Now, in this case, this is rather large. We can just zoom into this to really see what is happening in each step. And then what's really been important in this development of this tool, and what most people really appreciate here, is the ability to see example reactions on the right. So we have a proposed reaction here. We can see the nitrile being converted to this amide. And we can see now on the right-hand side the examples from the literature. And the examples are picked based on similarity. So the more similar a molecule is, again, we have a different scoring system here for similarity. The higher this number will be, and the higher in the example list it's going to be. And this gives you all the information for this example reaction that you would find usually in Reaxys as well. So the conditions, including the detailed procedure, if that's available, and the links to the full text. And if you want to explore this document a little more, you have those links available as well. One thing you'll see, I have this orange button here. I will unselect this just to illustrate what happens. What happens is you get the full list of examples, and for every step, you'll get 25 examples in decreasing similarity. So that really gives you a lot of literature review capability here to evaluate the feasibility of this step. Sometimes you'll find that this may be exactly what you need. You may find there are different ways of doing this in the literature, and you may resonate with some more than others. And if you do, you can select the ones that you want to keep and apply this kind of filter that way, and to reduce it down to these 2 examples in this case. And then you would just apply this process for every step. So we now go to step 2 in the retrosynthesis, and we're going to have a refresh of those examples specifically for our situation here. We're going to basically create a new heterocycle here out of these 2 compounds coming together. And there's some precedent for that as well in the literature that we can evaluate. Now, you will go through this process until you're satisfied with it, and you may find at some points, let's say if we go to the synthesis, if we look at this starting material, you have the option to again click on the shopping cart and take a look at what the suppliers say, or if you have your own internal database inventory, you can check if that's available. But maybe it's not available or it's not quite at the price point that you need. You have the option to extend your route. You can do that either here at the end. You could also do that in any place in the middle. If one of those middle steps you're not quite sure about, you want to evaluate an alternative, you can either click on these three dots and say add reaction step, or if you're here at the end, click on the plus. And what it's doing now is two things that are really useful. The first is, while we did just run a full prediction of this route, we did have as a starting material something that is published in the literature. So we find 8 references here from the literature with starting materials, at least for this one. This is quite a bit more available as the original and this one as well. So we may pick several of these examples and evaluate them next to each other. At the same time, we can also run another single prediction here. It takes a little time, between 1 and 2 minutes. It'll look the same way. I'm going to go ahead with the published steps, add those here. And you can see now that we started in this rectangle. This was route number 1. Now we're in customized number 6. So that gets a different color. And if you go here, we can see there's now 2 potential entries into this material from the published literature. In this case, we can show the conditions, 0.1. And this will be now, in this case, you won't get example similar reactions. You will get exact literature references because that we got this out of the published literature. And so that's the power of having Reaxys, the Reaxys database behind it. You can immediately see everything that has been published in this space. And then depending on your preferences and your understanding of the chemistry, you may decide to maybe kick one of these out. And you'll have a new set of, or rather a new pathway to get to your target molecule. You'll be able to export this either with all the data that we have as references and examples, or if you just want the examples that you selected, you can do that as well. And you can get that into your PDF or Word document, share it, keep it as a reference, whatever it is you need to do. And with this, I will just let you know that the molecule I submitted initially is now finished while I was talking. So you could go straight to the next project and evaluate that. And yeah, with that, I'm happy to listen to any questions and see what we can help with.
Thank you very much, Bart and Max. There are a lot of questions. All right, let's start with Bart. There are questions regarding, especially if, you mentioned about new generation or training the chemists. So, do you consider it important that new generation of chemists, while studying, should be able to be faced with problems of drug development, and how the experience you had could be implemented in education? I guess this question is about retrosynthesis, and if the young generation or new generation of chemists should be getting used to such AI-based technologies. What are your thoughts on that?
I would say definitely. I think this way we are going. I mean, we are now at very sort of at the front of that wave. I think this now has been commercially available, these tools. But I think this in maybe 5 years, you wouldn't even think about it doing maybe a quick search anymore. I mean, I actually use the RT tool a lot, even if I roughly know what I want to do, just because I can do something else for 10 or 20 minutes. And then I just get very spot on references. I have to do a lot of things in parallel. So I really like everything I can push to the machine and don't have to deal with. So I think this will really be how it will just look like in 5 to 10 years. That being said, if you are looking for a job in the next few years, you do not have to be an expert with Reaxys RT. You will. It's super intuitive, and you will learn this very quick once you start. But I do hope that universities will start implementing this as well in their curriculum.
Quite a lot of questions for you, Pat. So one thing which we hear as well is, I guess it's natural to feel a bit afraid of adopting predictive tools. Will they replace the chemist? Will they not replace? Will they take our jobs? Will they not take our jobs? What are your thoughts on this?
Yeah, I'm not scared. I really see this as it's a tool. It is not replacing any trained chemist. So um, you still have to be very critical what comes out. One of the examples that we had, for example, that I can unfortunately not show you this because it's many proprietary searches, but one of the proposals that it came up with was a Friedel-Crafts reaction on a pyridine, which essentially is a very nice idea, and all the references, they are very electron-rich aromatic rings, and this would work beautifully. But then we had an electron-poor pyridine. So even though for a machine this looks like two identical structures, you still need to use your chemical knowledge to say, well, that's likely not going to work. And that's just one of the many examples. And there are usually much harder problems to solve. So that's really how I position the tool in my daily work. Maybe I know I have a big molecule and I know how to make maybe the right hand side, and then I have a middle fragment which I want to couple, and then I know what left hand side comes on, and I focus my work maybe on the right hand side or the left hand side, which are very complex. But at least I can use the tool to make sort of the link apart and have them just these building blocks generated by the computer. So I think we'll just get more efficient if you adapt it well and not obsolete.
Perfect. Thank you. Max, a question for you. So the route that we saw in tree view, it's a very helpful view, and we would like to share that with our colleagues, etc. Is it possible to share it? And typically in
So
any of our usual chemist tools like ChemDraw or Marvin.
absolutely. So you have the option to either copy individual steps or the entire route into your favorite structure editor to then manipulate that for a presentation or share with colleagues that way.
Wonderful. I wish I had that during my PhD. Would have saved a lot of time drawing sequences of chemical reactions. Another question, perhaps, this is an interesting one from Manuel. Can you use the feedback and rating from the users, e.g., to tailor the predictions to the preferences in your company? Or can you make the retrosynthesis model follow the preferences in your company? Either of you can take it, but or Max.
Shall I start?
So at this,
Oh, go ahead, Max.
sorry, go ahead.
All right. So the tool is from Elsevier, so we cannot change that. But what we do, for example, we reach out as a company to our users and ask them, hey guys, what can we change? How can I make it better for you? Of course. And I have to relay that to Elsevier, or it's something we can fix on our side, like integrating building blocks. Search preferences we can talk about, like best practice sharing, saying, well, these things always work for me, and these maybe are super fast, and we can exchange on that. And I think what is very important is the building block libraries. So I didn't go into too much details there. Also, I don't know what I can share, but that is really the endpoint for the tool. So if you are creative with your libraries that you set up, you can steer the tool in a certain endpoint. So that is it from my side. I guess Max has a few things to add from the Elsevier side.
Yeah, the only other thing is with help, you can play with the parameters to direct it a little bit, and we've recently also introduced a functionality that allows you to, for a single molecule at a time, either define certain bonds you prefer to keep and not be disconnected, or you can direct the first step disconnection in a certain way as well. So there is some progress that's just happened in the last few months that we've implemented in that direction.
Perfect. Do we still have time? I believe. Hopefully, someone will let me know if we are reaching the time. One of the, in your view, either of your view as chemists, what do you think is the major or biggest contribution of such CASP tools? Is it creativity, ideation, like, come up with the wackiest disconnection idea, or, I'm happy to have bread and butter chemistry of amide bond coupling and Suzuki, as long as it actually helps me make the molecule.
Right. I think
from I'll let you speak in a second. From what I've heard from talking to a lot of people that have used it, it happens sometimes that you'll be genuinely surprised by a proposed route that it didn't pop into your head before you saw it there. And so that aspect is there. On a daily basis, the main advantage seems consistently to be just the time savings, really. Not having to look up each material, each step, because even for easier syntheses, you'll probably feel more comfortable first checking the literature, checking your suppliers and all of that. Having that all basically done for you ahead of time, that's where a lot of people really just save the time to focus on much more important things and more challenging and oftentimes interesting challenges.
Yeah, and I can only echo that exactly. And I think that's also really why we in discovery chemistry went for Reaxys, because it's really excelling at doing exactly that, just taking sort of the less difficult, maybe less challenging things off my table, so I can actually focus on the fun part of my job.
Perfect. No, thank you very much for this. I think in the morning talk, Mark Waller gave a very nice statement, which is, as for any human being, it's always a bit easier to recognize a good idea than to come up with one. So when you see a route, you're like, okay, yes. So thank you both for the wonderful talk. And we have a few more questions. We will get back to you as if they are not answered yet. With that, I would like to hand over to our next speakers, the team of Reaxys, to show some tips and tricks from the experts at Reaxys to help you make the most of Reaxys. Over to you.